Today, I'd like to discuss about engineering of the viral vector, which can be used for the development of different kinds of vaccines. I think I'll use this microphone here. So I put down here as engineering of a highly efficient recombinant viral vectors for the development of prime booster vaccines. And now, just for brief review of the vaccinology itself, we know that there are 3 different types of commercial vaccines available. The first one is live attenuated virus vaccine. That includes Sabin's polio, measles, mumps, rubella, yellow fever, smallpox, varicella zoster, and there are many, many animal vaccines using this strategy. The second strategy is inactivated or killed whole virus vaccine. That includes Salk poliovirus vaccine and hepatitis A, influenza, Japanese encephalitis, and there are many animal vaccines using the killed whole virus vaccine strategy. And we now have 2 subunit vaccines that includes hepatitis B virus vaccine using 22 nanometer particles of surface antigen, and also we heard about HPV vaccine, Gardasil, is based on L1 protein of human papillomavirus. So these are the different types of the vaccines. There are many other types of the vaccines being developed, including the DNA vaccine, the peptide vaccine, and also combination of all these vaccines. Today, I'm going to cover the recombinant virus vaccines. Yesterday, we heard about the flavivirus vector, and today I'm going to talk about the rhabdovirus vector. There are many viral vectors being developed, including the poliovirus, poxvirus, adenovirus, herpes alpha, retrovirus, flavivirus, and rhabdovirus. Today, I'm going to talk about prototype rhabdovirus. It is a vesicular stomatitis virus, and it is a negative-stranded RNA virus, and it contains about 12,000 nucleotides in genome. And there are 5 structural proteins. These are nucleocapsid protein N, P, M, matrix protein, G protein, and L protein. L protein, P protein is phosphoprotein, N protein a polymerase complex. And VSV has a very wide host range, which is very important as a vector because it's supposed to infect all mammalian cells as well. Actually, the VSV infects many insect cells as well. There's no pre-existing antibodies in human, so we have no problem with pre-existing antibodies. And there are very conveniently 2 distinct antigenic variants. The one is known as the Indiana isolate, and the other one is known as the New Jersey isolate. Now, it is a convenient system because the antibody raised against Indiana does not neutralize the New Jersey virus, vice versa. So that means that you can use the prime vaccine based on Indiana serotype, and then you can have a booster vaccine based on New Jersey serotype, so that you can have prime booster vaccination. And VSV replicates really in high titers. You can get the 3 billion virus particles, infectious virus particles per ml during the 10-hour of incubation periods if you culture the cell systems. And it can accommodate as much as 6,000 bases. So you have a total genome of 12,000, but you can include 6,000. So you can have 18,000 bases, literally. And then VSV, recombinant VSV looks very long once you insert these genomes. And the level of inserted gene products is very high. So it's a very attractive system. Now we heard about Ebola virus vaccine based on recombinant VSV. And this Ebola virus vaccine based on the recombinant VSV was constructed with wild-type VSV. And that has some reactogenicity. And it has maculopapular rash and also vesicular dermatitis. And also, some vaccinees experienced fever, and some long-term effects was arthritis. So no matter how good the system is, it has to be safe and also not neurovirulence. And we are now trying to develop the vector system. It is totally safe for the human use. And using the priming vaccine vector has to be antigenically distinct from the boosting vaccine vector for the effective immunogenicity. And I think you would agree that. So we have a VSV Indiana priming, and we have VSV New Jersey priming with the same kind of the genes inserted into it. Now, we learned over the years that matrix protein of VSV is responsible for pathogenesis. And actually, that kills cells once you infect VSV in a wild type case. Now, if you change this matrix protein, actually single amino acid change, 51 position methionine change to the arginine, that virus become avirulent. It does not kill cell. In addition, the Enflammant in France, long time ago, isolated a virus which is known as a TSO23. It's a temperature-sensitive mutant which contains mutations at the 21 position glycine to the glutamic acid, and the position 111 as a leucine changed to the phenylalanine. And these two changes makes this virus temperature sensitive. So we combined this asymptomatic or non-cytolytic virus mutation from M51R with these temperature sensitive mutation. So we have G21E, M51R, L111A. Now, instead of phenylalanine, we change it to the alanine because we found that if you have single nucleotide change, 20 consecutive passages of this virus really makes conversion of this back to the TTG, that is, to the leucine. So in order to make this virus or mutation stable, we changed all triple codons. So we make, instead of phenylalanine, we change it to the alanine. And this virus is completely stable. You can make 50 consecutive passages with high multiplicity of infection. It still stays at leucine, change it to the alanine. So that's very important. And this one, we call it VSV Indiana GML. In addition, we also made a mutation of the M51R and M48R. This is a New Jersey strain that contains the 2 methionine residues in a span of 3 amino acids, and we changed this M51 methionine to the arginine at the position 48 and 51 to arginine. And again, we change all 3 nucleotides so that it does not convert back to the wild type. In addition, we also introduced similar mutations at this site. And if you change leucine to the phenylalanine at the 110 position, and then leucine to the alanine at 110 position, this becomes also partially temperature sensitive. So we have these 2 vector systems using 2 different the serotypes. Now, it is possible using the so-called the reverse genetics, we can recover all mutated RNA genomes by using reverse genetics. And we have cloned genes which contains T7 RNA polymerase promoter, and then also hepatitis delta virus ribozyme. And when you co-transfect this cell expressing BHK T7, T7 RNA polymerase, and then in addition to the RNA polymerase complex which I talked about, this will be transcribed, and the RNA polymerase complex of the VSV will replicate this genome, and you can have a progeny virus produced. So this is how you can recover the virus. So you can introduce any type of mutation and recover as a recombinant virus. And doing so, we have isolated these mutants and look at the temperature sensitivity of these viruses. As a wild-type virus grows almost to the 3 times 10 to the 10 to the 9 rather, 3 billion virus particles or PFUs per ml. And when you incubate at the 37 degrees, there is about 3.5 log reduction in terms of the virus replication with the VSV Indiana GML. And then the VSV New Jersey GMM with the isoleucine changed to the alanine, that drops about 1.5 log. So these are the temperature-sensitive mutants. We look at the cytotoxicity of these cells after infection, and it turns out that the GMM and the GMM isoleucine to the alanine change makes there's no cytopathic effects. And the same here at the 31 degrees, there's a little cytopathic effects. So the in vitro studies show that there is less cytopathic effects with M mutations. And we look at the expression level of the virus proteins, and actually it doesn't matter whether it's a 31 degrees or 37 degrees. 37 degrees is a semi-permissive temperature, and 31 degrees is a permissive temperature. At both temperatures, we see same level of VSV production. In other words, TS023 is assembly defective virus, and the protein expression level are the same whether it's at the 37 or the 31 degrees. And then we look at the neurotoxicity of this virus, and by injecting mice intracranially, we can actually purchase a mouse with a little hole on the skull of the mouse, and then you can inject the virus as much as you want. And if you inject the wild-type virus as little as 1,000 particles, all mice die within 4 days. Whereas if you inject those mutants, they all survive. Even if you inject 1,000 times, these are non-neurotoxic. This is Indiana, and the same with New Jersey. New Jersey is less cytotoxic and less neurovirulent, and we see one mouse actually paralyzed, but other than that, it's less neurotoxic. However, when you change this to the M mutant, both M mutants show that there's the perfectly normal growth. So we now have the VSV, the Indiana and VSV New Jersey with mutations in M, and Indiana we have the triple mutations. It's the glycine to the glutamic acid at the position 21, methionine to the arginine change at the position 51, and the leucine to the alanine change at the position 111. And this is the priming vaccine. And then we boost with the VSV New Jersey with the GMM, and which contains the G21E, M48R, M51R mutations, and as a boosting vector. The reason that we are using GMM instead of GMML is because the titer of the virus is higher with GMM, and it shows no neurotoxicity. Therefore, we are using GMM. Now, we want to test whether or not that this is effective vaccine vector. So we have included the HIV genes of GAG, POL, and M genes and inserted into the VSV. Now, the insertion site is at junction between the G and L genes, and we put entire GAG gene, entire POL gene, and entire M gene, put it into here. And also, we combined GAG and POL together. As I told you, that it can accommodate as much as 6,000 bases, and we have done the expression of hepatitis C virus genes. If you put all non-structural genes, like NS3, 4AV, and 5AV, it's above 6,000 bases, and it can express all the proteins. So this is absolutely no problem expressing GAG and M together and the POL separately. Now we look at the level of expressions. These are the empty vectors of the New Jersey, and this is empty vectors of the Indiana and New Jersey, right? Indiana and New Jersey. And when you add those GAG genes, you can have expression of P55 precursor proteins of the GAG. And P55 unprocessed GAG protein forms virus-like particles. And that is shown here, that when you infect those cells with the recombinant VSV carrying the GAG gene and M gene, they form virus-like particles. It's about 100 nanometer particles, but it's an empty particle, and it represents very good antigens. Now we look at the expression of M genes, and we look at the GP160 precursor protein for GP120 and GP41. And also we look at expression of the pol gene. The pol gene is heterodimer of 51K, 51K and P66, and this is cleavage products. Then we look at the antigenicity, immunogenicity of these. And so we have the VSV Indiana, GML carrying the HIV gene, and we use this as a priming vaccine, and then VSV New Jersey, GMM, carrying HIV genes as a boosting vaccines. First, look at the CTL responses, and this is the vector alone, and here with the GAG gene inserted. This is a single peptide that specific the CTL response. And this was a big surprise because we see as much as 20% of the total CD8 positive cells were reactive with this specific epitope. And we have the pol gene products and also EMV, and this is a combination of GAG, EMV, and pol. Now, just remind you that MHC class 1 mediated cytotoxic T cell response is based on the activation of CD8 positive T cells. So you can activate the CD8 positive T cells by recombinant VSV, and that will express viral-specific antigen. In this case, it's HIV-specific antigen, and that activated T cells can recognize MHC class 1 and viral-specific peptide conjugate. Actually, this is the receptors, the target for the T cell receptor. And what happens is that virus-infected cells can express the viral-specific proteins that is cleaved by proteasome and that is entered into the ER and combined with MHC class 1 and goes out of the cells. And this complex here, MHC class 1 with a beta-2 microglobin with a peptide, the viral-specific peptide, is a target for the T cell receptor. And then this is destroyed. In other words, infected monocytes and macrophages, as well as dendritic cells and T cells infected with HIV, can be targeted with educated CD8 positive T cells. Those are the CTLs. And in that case, we can really destroy this cell by apoptosis. So that's the whole idea. The second immune response that we looked at is the antibody response. This is the humoral immune responses. As I told you, that the GAG precursor protein forms virus-like particles. And these are secreted from virus-infected cells. Therefore, it does trigger the humoral immune responses to make antibodies. And so the GAG P55 produces good antibody levels. And also, the GP120 is a secretory protein, it's a glycoprotein, and since it's a secretory protein, it too produces or triggers humoral immune responses. So we have antibodies made against GP120 and GP41. So in summary, the genetically modified MG mutants of the VSV Indiana GML, those are the P51 actually methionine changed to the leucine, arginine, and then there's the mutation at leucine at the 111, position 111, to the phenylalanine. In this case, it's alanine. And that mutation, and also at the position 21, the glycine changed to glutamic acid, that really is a temperature-sensitive mutant, and that's effective in producing or expressing the foreign genes. And these, the mutants, can produce as much as 3 billion infectious particles per ml when you infect the cells in culture. And now the GMML, which we found that it is a temperature sensitive, but this virus is about 50% lower than the other two viruses, and so 7 times 10 to the 8 PFU per ml. And therefore, since neither GMM mutant or GMML mutants are neurotoxic, we decide to use the GMM because it produces higher titer of the virus. And these recombinant VSV vectors are very stable and avirulent. And VSV Indiana GLM priming with GMM boosting do not cause any adverse events and induce robust adaptive immune responses. We have tried to immunize with New Jersey first and then boosting with Indiana. That was not as effective as priming with Indiana following with New Jersey boost. The genetically modified MG mutants of VSV, GLM, and VSV New Jersey GMM prime booster vaccine vectors are applicable for many viral as well as bacterial diseases. We are talking about there are many, many different diseases that we cannot control as yet. And we are talking about the MERS, SARS, we have the other dengue viruses, and even bacterial disease such as Mycobacterium tuberculosis. The P55 gene may be inserted into this vector and make second generation tuberculosis vaccines. And so those of you who are interested in this vector, it is available, and you can contact me. And this system may not require cold chain for distribution, which is very important. If you have a vaccine, if you want to distribute in developing countries, and the cold chain is very important aspect. So this can be lyophilized, and then you can reconstitute with the cell line, and then inject people this way. So this system is a highly efficient system for the development of the vaccine, which can trigger not only the humoral immune responses, but cellular immune responses. With that, I'd like to acknowledge my group in my lab, and also the company who is sponsoring our clinical trials of HIV vaccine. I'm not talking about that yet, but we have the clinical trial going on with the prophylactic vaccines. And the research is sponsored by the CIHR, which is Canadian Institute of Health Research, is equivalent to NIH grant. And the National Research Council, the NHRDP, is the National Health Research Development Plan, and also the commercial company known as Sumagen Canada. And I thank you very much, and I'll be happy to answer questions. Thank you.